From Microbe TV, this is Immune, episode number 20, recorded on May 28th, 2019. I'm Vincent Racaniello, and you're listening to the podcast about the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Good afternoon.
Are you in winter yet?
Yeah. What?
Just wanted to confuse you.
I went kayaking yesterday. It was lovely.
You could kayak in the ice and stuff, right?
No.
Where do you kayak?
On the lake. Cayuga Lake.
Lake Cayuga. Cool.
Yep.
Also joining us from Durham, North Carolina, Steph Langel.
Hey there. Things are good here. I'm sweating. It's like 97. So I'm adjusting from the Ohio to the North Carolina weather. So people have told me just to buckle in because it's going to be like this till maybe September. So.
90 degrees till September, right?
Yeah. I don't know if it's as bad as Florida. I think Florida gets the humidity and heat worse. But, I'm taking it in.
You'll be there for a few years.
Yeah.
All right. Well, here we are again a month later, and we have some science for you. But I want to just remind everyone first, if you enjoy what we do, consider supporting us financially. You can go to microbe.tv/contribute. And there are a couple of ways you can help us out with our expenses, etc. We'll have more about that later. And it is Cindy's turn.
It is.
Yay.
What's new, Cindy?
What's new? Well, I went to the American Association of Immunologists meeting a couple weeks ago, and that was really fun. First of all, we have fans. So there were a couple of people who came up to me and were like, oh, you're Cindy. Oh, yeah, we love Immune. So that was great. So we're getting out there.
Where was the meeting?
In San Diego. So that didn't hurt either. It was very nice.
Right.
Yeah,
and next year, I think it's in Hawaii. Is that correct?
Yes, it is.
Yes. Well, I may decide I didn't have enough data this time, so maybe next time.
They give out a lot of money in travel awards. So I highly recommend, if you are a trainee, to join the AAI and apply for those travel awards because they're really great.
That's great. And maybe we can work. I mean, what if that was a Destination Immune episode where we could get Vincent out there?
That would be great.
That would be great.
Plant the seed early, right?
Yeah. Right.
Yeah, Hawaii's a nice place.
Yeah. So there was a lot of really cool science there. And one of the things that a couple of the talks were on was the idea of the intersection between coagulation and inflammation. And it's something that has always, nagged at me in the back of my head, but not very many people research this topic, but I think it's probably going to be one of the hot things going forward over the next few years. So I think there's a lot to learn. And so I thought it would be really cool to do a paper about just this topic. And I kind of picked one that was a little closer to what I know, so that would be a little easier to get through, because I don't know the coagulation cascade, so we might have to tap Vincent for that, since he went to med school and learned that at some point in time.
Me.
I got a graphic in here in the notes, so if anything, we'll just look at that.
Yeah. So, but our lab has started to dabble in that too in a collaboration. So it was kind of fun to read that in a little bit more detail. And so the title of the paper that I picked has a lot of words in it, and we'll explain them. But I thought it's cool to introduce a lot of these topics too that we haven't talked about. So it's called Inflammasome Activation Triggers Blood Clotting and Host Death Through Pyroptosis. And it's by a number of different groups, mostly from the University of Kentucky, National Institute of Biological Sciences in China, UNC Chapel Hill, down by you, Steph, Tongji University in Shanghai. Also, Genentech is making an appearance, and National Institute of Genetics, Mishima, Shizuoka, Japan. And the lead author is Kongqing Wu, and the senior author is Zhengyu Li. Did I do a good job of that?
Sounds pretty good.
Yeah, it's pretty good.
Not bad.
I think people can correct us if.
Yeah. But yeah, so I thought that we would start by introducing a couple of the players here, because these are some things that we haven't necessarily talked about. And the first one is the very first word in the title, which is inflammasome. And so inflammasome is not just one thing, but a bunch of different things. And I'm interested in innate immune recognition of microbes, and with a primary focus in my lab on surface receptors like toll-like receptors. But these are molecules, protein complexes that are expressed in the cytoplasm. And they're there to see things like viral infections and bacterial infections. And so a lot of these inflammasomes have an initiator protein that recognizes some microbial structure. And it's often, in this case, bacterial products. And then it assembles quite a beautiful structural complex of a couple different proteins, including an apoptotic spec protein, or ASC or ASC, and then a caspase. And so a caspase is an enzyme that will cleave another protein. And so the main proteins that these cleave are something called IL-1 and IL-18, which are cytokines. And so IL-1 and IL-18 are some of the most inflammatory cytokines that we have. And if you follow this, there's an inherent problem here, because these protein complexes, these inflammasomes, are in the cytosol. They recognize bacterial products in the cytosol. So bacteria can get in the cytosol. Either they can invade and replicate in the cytosol, or there are also bacteria that express proteins like a needle protein, like a type 3 secretion system, type 4 secretion system. And when they bind to the surface of a cell, they inject in proteins and mediators to modify the cell. And these inflammasomes can recognize some of those proteins that get injected in, and that will become important here. And so this is a defense mechanism that can be expressed by any cell, pretty much, although which inflammasome components are expressed in which cells can be more or less specific, but technically any cell can have this capacity. And so they assemble these complexes into these beautiful structures, and they cleave this IL-1 and IL-18. And the problem is these are cytokines, right? Cytokines are secreted signaling molecules that communicate between cells by being secreted by one cell and binding to a receptor on another cell. But these are produced as cytokines in the cytoplasm, and they're cleaved by this inflammasome in the cytoplasm. So how do they get out? And this is something that has bothered me for a very long time. And there have been a couple of papers a while ago that said maybe some LC3 mediated autophagy mechanisms might internalize the cytokines and then secrete them out of the cells. But there's another mechanism that's come out more recently that's called pyroptosis. And there's lots of different apoptosises that have come out: apoptosis, pyroptosis, necroptosis. So there's like many, many different ways that cells can die. But this particular one is highly inflammatory. And that's really different from what we typically think about of a cell dying. Usually we put it into two camps. Either a cell kills itself in a very quiet manner to package itself, either during development or at the end of an immune response or as a mechanism to shut something down, or cells are traumatically damaged, burst open, and release a lot of inflammatory mediators. Well, it turns out that there's other forms of death that can be induced, but still highly inflammatory. And this pyroptosis is one of those. And so there's a lot of different proteins that are involved in this process, but the basic idea is that there is activation of proteins that form pores in the plasma membrane and allow the IL-1 and IL-18 to get out. And so this is one way in which these highly inflammatory cytokines, their production can be regulated or their release can be regulated. You need something really damaging that's going to induce inflammation, and the cells know that they really need to produce these IL-1 and IL-18 because they're so damaging.
Right. And a couple things. One, if you're a visual person, you should really take a look at what these inflammasomes look like. It kind of blows your mind, the amount of things that are inside cells, particularly in the cytosol, but it's like this wheel-like structure. When you're looking down on top of it, actually, those ASC proteins that kind of come down off the bottom. But when you look on top, it's just like this really pretty wheel-like structure. And then, the other thing is, thank goodness we have all these different ways for cells to die, either programmed or induced, because imagine all of our cell turnover. I mean, if that wasn't nicely and quietly done, our bodies would just be constantly activated in this inflammatory state, which people who have, mutations, some of these genes, that's actually what happens in these autoimmune diseases. So yeah, inflammasome, this innate immune stuff. I'm more an adaptive immune person. So I love kind of learning a little bit more. And it's amazing kind of the complexity, but yet simplicity of these different ways that cells can die. Speaking of virology, I mean, it definitely touches upon Vincent, in viral replication and cell death. It's a good crossover for you as well.
Yeah. It's another way that cells die. When they're virus infected, all these toses that Cindy mentioned come to play in virus infected cells, for sure. I'm thinking about something you said, that there's so many ways to die because otherwise we'd be inflamed all the time, right? Isn't that what you said?
Well, if we didn't have a way, if apoptosis didn't work, so that inflammatory danger signals inside the cells weren't easily packaged and inside maybe vesicle and then dealt with by surrounding cells, your body would react to those, which really is what pyroptosis is all about.
Most of the virus action is in the other sensors, but this one is also involved. It's interesting. I don't know why, if it's less important or just hasn't been studied for viruses.
Yeah. And Cindy, maybe you can touch on it more, but even with this paper we're going to go over, I guess I was surprised we didn't know this already. Not being in this subset of immunology and this, particular field of immunology, I just was surprised. But Vincent, maybe you're right. Maybe, they talk a lot about bacterial products in this way of pyroptosis. But the intersection of what viruses are up to with inflammasomes, I'm sure somebody's looking at that.
Yeah, there are papers for sure. You can find them.
Right.
I was looking at one involving rotavirus the other day from Harry Ginsberg's lab, for example. Mm-hmm. So it's out there.
Right. Yeah. It is. Yeah. So anyway, it's interesting that there is this specific mechanism of cell death that can then release these cytokines. And there's also one more that I think it's called necroptosis that people use, because there's these things called necrokines, which are similar to IL-1 and IL-18. These need to get processed and released, but so necrokines are only released when the cells burst open. And so it's really a damage cytokine, indicating that there's a lot of tissue damage going on. So there's lots of different ways for the body to communicate, I would say, danger, right? It comes back to the danger hypothesis, controlling release of these highly inflammatory cytokines to when they're really needed due to these multiple inflammatory or damaging signals that come. And to produce this IL-1 and IL-18, remember, you still have to make the protein too. And so this is another one of those backup or, I don't know if you have, I don't know how I want to say it, but it's like you have to have this 2-step process to make these and then release them. And so TLRs are a main way to activate production of IL-1 and IL-18, but then they sit in an inactive form until they're activated by the second signal, which then allows them to be cleaved by this inflammasome process. So the most important takeaway is this is a really controlled process, a very precise cell death in an inflammatory way that we hadn't, it's very well described now, but it's definitely not something we were thinking about a number of years ago. So the other really cool thing that we're going to talk about is coagulation. And I'll tell you right straight off that this is not my forte of things. I still don't understand really the the coagulation pathways. And when I've taught this, and I'm going to be honest out there, when I've taught this in the past, I'm like, okay, there's kinins and there's bradykinins and there's factor blah, 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 7 and 10 and this, that, and the other. And it's all really important for connecting with inflammation. And that's about all I've ever said when I teach immunology, because I didn't understand it. It's really complex. It's really in the camp of the hemostasis people who study this. And very few people look at that cross-section with inflammation. Right. But the reality is that the lower organisms and insects and things, they use this as a main mechanism of defense. So right. they wall things off. They form, maybe it's not what we could call a traditional clot, but they basically clot around things and wall them off. And that's a defense mechanism. And our defense mechanisms have evolved so much from that, and they're so complex that we forget that sort of primal defense mechanism. And if you think about it, this is I study innate immunity, and we study things that happen really fast and pre-made factors and things. And we're talking minutes to hours of cells being recruited and activated. Coagulation is seconds to minutes, right? As soon as you have damage and you have exposure to the proteins that are important for coagulation, it starts. And so this is really the first defense and the fastest defense. Yet we don't study it that much in the context of immunology.
Right, right.
What do most people study it in? What context?
It just in hemostasis and blood regulation of blood. I would say that the one place that people have looked at it more from a medical standpoint and less from a mechanistic standpoint is in sepsis. Mm-hmm. Right? It's certainly not the only disease. There's clotting. We now know that if you have cancer, you have a higher probability of having a thrombotic event. But the most well-studied, I think, is in sepsis, because the main thing that kills people that get bacterial infections in the blood and sepsis is disseminated intravascular coagulation, or DIC. And basically, that's lots of little clots that happen all over your body. And they cut off circulation to organs, and you end up with multi-organ failure. And that's how people die of sepsis. And so we say that, and it all makes sense, and it's like, okay, so yeah, bummer that you get these little clots all over your body, and it's really inconvenient, and it shouldn't happen. But if you think about it as a defense mechanism, it becomes really intriguing, right? So what if what we have thought as a very inconvenient thing to happen is getting these clots all over your body because of hyperactivation and bacteria in the blood? What if it's actually to try and fight the bacteria? Right. That you're trying to form clots around those bacteria because the sepsis is so dangerous and the immediate problem that's the best and fastest way to solve it. But if it's not cleared rapidly, then you have death, right? You get these secondary events. So it's just it's been out there, but it's just not something that I've thought about that way. And I think, wow, that's really interesting. That's really cool. And before we get into the paper, there's a couple more things. And one of the things was there was a presentation at AAI where they talked about the macrophages in the peritoneal cavity, and that they have coagulation capacity, and that when you expose them to bacteria, they form little mini clots. And so if you have bacteria and macrophages floating around in your peritoneal cavity, for them to find each other is challenging. But if the macrophages start to form a clot and capture the bacteria, they can now find them. And so it's sort of like that idea of neutrophil nets. When you cast out those nets to trap the bacteria, we think maybe the macrophages, at least that's one cell type that can do this, can cast these clots basically and capture bacteria so that they can try and eliminate them. Anyway, I just thought that the whole concept of this was really, really interesting.
Oh, that's great. And so when you were at AAI, were you drawn into, because there's so many things going on at once and you have to pick and choose which sessions you're going to go to. Were you drawn to this because innate immunity, and then they were talking about coagulation, like it just happened to be in the same session?
So there was a monocyte session and there was a whole bunch of things, and that was one of the talks, and then there was a couple more spattered out through other sessions that I was in. So it was spattered out all through the meeting in different sessions and things, but it just seems to be starting to come up. It's on people's radar as something interesting to think about going forward. So that was neat.
Great for an immune.
Right, exactly. And that's what I thought. I was like, well, let's pick a paper in this topic so I can learn more about it. But so how do you induce coagulation, right? So the primary way is if you have vascular damage, you have exposure of a protein called tissue factor. And so it's normally held on the non-exposed side of a vessel. And when the vessel is broken, the tissue factor is now exposed. And when it's exposed, it triggers a cascade of events that basically lead to cleavage of a protein called fibrinogen, and that deposits fibrin, which makes the clot and stops the bleeding. And so all of that's related to stemming the blood loss, right? Because what's the number one thing that happens if you cut yourself? You want to stop bleeding because that will kill you first before anything else, whether or not you got bugs in your, but introduction of microbes, right? We've got to stop the bleeding. So that's number one. But then it's interesting that all these things are connected together, because now we're starting to understand that tissue factor is playing a role in inflammation as well, and inflammation can regulate tissue factor. So it's back and forth between those two things. And then the last player that we need to introduce is microvesicles. And I can tell you that, again, I'm not the expert in microvesicles, but these are little packets of communicators that communicate between cells. And so in this paper, what they found was that there are microvesicles that carry tissue factor. And so they get released, and that can contribute to the development of coagulation. And so although there's a bunch of different papers, this is one of the first ones to show this connection. It's been simmering for years, but this is a nice way to sort of show mechanistically that clotting and inflammation are connected, and they're connected through this process of pyroptosis and release of, in fact, microvesicles through that process.
And I wonder, so the microvesicles, there's many different sizes and shapes. There's like exosomes, there's so they all kind of fits under this extracellular vesicle category.
Correct.
And they don't really go into that too much, like defining the microvesicles. I was kind of interested a little bit more into their, how big they were, what else was in there. They don't really go into that, but it does open the door to a whole another aspect of this and how the cells are communicating, in this case, the tissue factor, to other parts of the body. So I did a seminar recently on, there was a paper that came out that looked at rotavirus and norovirus being able to have an enhanced transmission if they were packaged in microvesicles, and the size was dependent on the virus. And so that kind of piqued my interest. I had just read a bunch about that. So yeah, it's another little aspect to this paper, is thinking about, not only these inflammasomes inside the cytosol, but then how they're being, well, I don't want to say blebbed, because blebbing is a different thing, but how the microvesicles are coming off the plasma.
Yeah, so there is different ways to get microvesicles. So some can bleb, some get internalized into like exocytic compartment and then secreted out from within that compartment. And then I guess others can be made different ways as well. So, and they each carry different things, but they can carry RNA, they can carry DNA, they can carry microRNAs, they can regulate gene expression. So it's a really interesting area of stuff that's coming.
It's a wild world in that cell.
So a microvesicle is a kind of extracellular vesicle.
Right.
That's the kind of broad name for everything, which includes microvesicles and exosomes, right?
And I think there are some other ones too.
They differ by their size and what they contain,
and how they come out. So exosomes are going to fuse with like a lysosome inside and then kind of release the contents, whereas the microvesicles are more taking like pieces of that plasma membrane with it as it comes off. Yep.
So lots of cool players in this paper.
Yeah.
And so the last thing that I have to introduce is, so to activate this inflammasome, you need something introduced in the cytosol. And so some bacteria have these type 3 secretion systems, and they inject something into the cytosol. And in this case, in this paper, they are using a protein called EPRJ. And I don't remember exactly what that stood for.
Yeah, it's not rolling off the
I
top.
don't know if they defined it, actually. I was looking for the thing. Anyway, so they express this protein in E. coli. And since it's not being injected in the way they're using it, because they're using just the protein, so they're eliminating using the bacteria or the secretion system or anything, so they can isolate this player in a reductionist way and look at what it's doing mechanistically. So in order for it to work, it has to get in the cytosol. And proteins don't normally go into the cytosol, right? So they get injected in if the bacteria has type 3 secretion system, but in this case, they're not using that. So they make a recombinant protein. So how the heck do you get the protein into the cytosol? So in this case, they did a cute little thing where they fused it to anthrax lethal factor, which actively binds to surface receptors and penetrates into the cytosol. And so it allows delivery of this EPRJ protein that's now fused to anthrax lethal factor to get into the cytosol and activate the inflammasome. And so one of the first things they did was just see if that works. So can they add this to cells, and can they induce this activation of the inflammasome? And they find out that does happen. So they can activate the cleavage of caspases and other factors in the cytosol that allow them to conclude that the EPRJ protein is inducing this pyroptosis, and it's activating caspases and, has these effects. So then what they really want to do is show that this is going to work in a mouse. So they use a mouse model. And so they inject this fusion protein into mice and ask, does it do anything? And one of the first things they looked at was whether it induced lysis of red blood cells. And it did. And one of the things that is indicative of is activation of a coagulation cascade. So sometimes the red blood cells get lysed. And so they specifically went and measured a number of parameters that occur during this sepsis-induced disseminated intravascular coagulation, or DIC. And so they measure several different parameters that are associated with the development of coagulopathies and DIC. And these include something called prothrombin time. And so the prothrombin time will go up. So it takes longer to cleave thrombin in vitro if you've used up all the factors that are necessary. So if you take a mouse and you bleed it after you've given it a treatment, and you measure the prothrombin time, if they have a lot of their thrombin activating factors present, they will cleave the thrombin very quickly, and it will have a very short prothrombin time. But if all those factors have been depleted because they've been used because they're inducing clots everywhere around the body, now it takes longer to activate the thrombin. And Steph, you put a beautiful thing of the coagulation cascade in there. We could talk about it a little bit, but again, I'm not sure I really fully understand it. But there are two ways to activate this pathway. There's an extrinsic and an intrinsic. And so tissue factor is involved in the extrinsic pathway. So when tissue factor gets exposed, it will activate factor 7, and then that feeds into factor 10. And that cleaves prothrombin into thrombin.
Right. If you're by a computer listening to this, it might be beneficial to pull up your own coagulation cascade just to follow along, which the one that we have in front of us, the tissue factor, I think we'd have to modify it after reading this paper. Right. And I'll explain a little bit about that
because it can be
once
released,
we get to the end.
Yep.
Right. Right.
Yep. Exactly. So needless to say, they have this increased prothrombin time. Another thing that happens is I mentioned that when you have a blood vessel break and you have to clot right away, you cleave fibrinogen into fibrin, and that fibrin forms the clot. So the amount of available fibrinogen in the blood is an indicator also of whether or not you have ongoing coagulopathy and clotting going on everywhere. Because if the fibrinogen is at low levels, it means that most of it's been converted to fibrin and is in clots around the body. And so after injecting the mice with this EPRJ protein, they then looked at fibrinogen, and it was quite low compared to the control mice. And lastly, they look at alpha thrombin and thrombins. This is called thrombin alpha thrombin or thrombin antithrombin. So this is TAT measurement. And again, that's really high the more active thrombin you have. So the more coagulation that's occurring, the more active thrombin you have. And so the higher that level is. And that level is really high. And so as a consequence of these increased coagulation parameters, you'll often have loss of platelets. And so again, they measure that as well. Within the first 90 minutes, there's a loss of platelets. And they see that, and this is important for the rest of the paper, is that they are able to isolate microvesicles from the plasma of these mice that have been injected. And they have lots of microvesicles that are filled or decorated with tissue factor, have tissue factor activity. So altogether, this means that when this protein that we know is supposed to activate the inflammasome happens, and it happens in the body, all of a sudden you now have all of these thrombotic related events or coagulation related events in response to that. And really, we've looked at what is the immune response related to injecting those proteins and not this effect on coagulation. But it's saying there's really a connection between the detection of the microbe via these inflammasomes and induction of this procoagulative activity.
Right. And I think, figure one, they really do a good job of convincing you that, this EPRJ does induce systemic coagulation. And, it's interesting because all of these different things, they do describe that they use this for this DIC, the disseminated intravascular coagulation diagnosis in patients. Now, I don't know whether they just do the prothrombin time or if they do all four of these, A through D, but that was pretty neat. And then the other thing I was happy to see was, you had mentioned that to get that EPRJ through the plasma membrane into the cytosol, they have to combine it with that lethal factor, the anthrax lethal factor. They do confirm that in combination with a little helper protein does not itself cause anything. I think that was in the supplementary to kind of help convince you that, yes, we're using that to help get it through the cell, but it's not by itself causing any issues. So
that's right.
I was glad to see that.
Yeah. They also did a couple of really cool things where they showed this visually, and I'm a visual person, so I love this. So they were able to do intravital microscopy of the cremaster muscle in the mouse after they injected this EPRJ protein. And what they could see was microthrombi in fibrin deposition. And so I just thought this was really cool because what they did to do this was they harvested and purified platelets from GFP expressing mice. So they have green platelets, and they injected them to a mouse that is not labeled. So it's just a normal mouse. And they also inject in anti-fibrin antibody that's fluorescently labeled. And then when they inject the EPRJ protein, all of a sudden now they see green cells trapped in red positive clots within blood vessels in live animals. And I thought that's really cool.
Very cool.
So cool that they could do that. So it's pretty convincing that there is this coagulation that's induced. And since this characterization of this response to this particular protein has already been described, this is saying that, there is likely a connection between this inflammasome and this coagulation. So is it and how is it occurring? So that the rest of the paper is on this mechanism part. And so they use mouse models to ask this question. So again, throughout the whole paper, basically, they're just measuring their prothrombin time, their fibrinogen, and their TAT and survival, and some cleavage and tissue factor expression. But generally, those are the markers. They're just reading out these DIC markers. They're asking if the mouse lacks the ability to activate the inflammasome, what happens? And so this is also an interesting thing for those who are immunology aficionados. So people have been using a caspase-1 deficient mouse for years. Turns out it's both caspase-1 and caspase-11 deficient, which creates a whole lot of confusion about this. It was discovered a number of years ago, but you have to be really careful if you're going to try and look at what role caspase-1 is playing in any particular process. And so luckily they also have caspase-11 only knockout mice, so they can look at what is attributable to both or to one. And so what they found was that although the prothrombin time goes up and the fibrinogen goes down and the plasma TAT go up in a wild type animal that is injected with this EPRJ protein, that if you lack caspase 1 and 11, this doesn't happen. So that suggests that you absolutely need this formation of this active inflammasome complex in the cytosol in order for this to happen. And if you use caspase 11 knockout mice, nothing. And if you use TLR4 knockout mice, nothing. It's the same as wild type, which I guess is important because if you're making recombinant proteins in bacteria that have LPS, there's LPS, you can trigger TLR4,
That's true.
et cetera, et cetera.
Yeah.
So nonetheless, it's happening via this protein through some sort of inflammasome process, and that this is really important for activating all these inflammatory sequelae. The other
interesting. Oh, sorry.
Go ahead.
You can go ahead. Well, for this figure, they were talking, so they have the caspase 1 and 11 knockout mouse. They have the caspase 11 knockout mouse. What about the caspase 1 knockout mouse? They actually describe that they have one, because it's challenging to get
it is
what they described as clean, which, I don't know if they created that mouse by doing like a lot of back crosses, or if there's some other ways I think you can use like the black 6 embryonic stem cells to help generate that, but they don't really include that in the figure. It's kind of hidden in the supplemental, but I think it's a robust way to show that caspase 1 looks the same as the caspase 1 and 11 knockout, meaning it is caspase 1.
Right.
I don't know why they didn't include that.
I don't know. I'm wondering if maybe they're not very healthy mice, and they're hard to come by. I don't know.
Yeah, that was it, because that further proves that it is caspase 1.
Yeah. So I think it's pretty clear. I think it's pretty clear. It's caspase 1. The other thing they did that was really cool is that if you inject this protein into mice, they will die. They, because they get this disseminated intravascular coagulation. They showed that, and they will not survive this. But if they lack the caspase 1 and 11, that particular model, they survive this. They don't get the DIC. So, one, that's really interesting because now you have a way to potentially prevent the DIC during sepsis, is not necessarily targeting the coagulation cascade, but targeting the recognition of the microbe to begin with. Because they try to treat by lowering the amount of sepsis, treating with antibiotics and things, and maintain blood pressure, try to, put them on antithrombotics. But maybe that isn't going to work, because if you still have recognition of the microbe and release of these cytokines, it still may induce the sequelae. And so you might not be able to easily inhibit it.
So what is the relationship between this assay where you put a protein in, which gets into cells, EPRJ, and having whole bacteria? Say you gave the mice sepsis, actual sepsis. The assumption is that this protein would be injected in. And so why not do it with bacteria and not just this protein?
Well, that's a good question. I think it would be complicated because you would have other innate mechanisms to recognize them, and then you wouldn't be able to, at least in the whole in vivo model, isolate that to the recognition through inflammasome is causing the coagulation.
Right.
Because we already know that bacteria can do it.
Yeah.
Right. That's true. Maybe it would have been good, though, just in the beginning to say, okay, here's the big picture. Look what's happening, to make sure that there's that association. But you're right. I mean, it doesn't get down to the memechanism, but
I think it would be interesting to do that initially and then say, take bacteria lacking EPRJ and see how it differs. Right.
I don't know I don't know if they require it, because I don't know. Again, I'm not really sure if I know which protein, what it does. Maybe one of our listeners can shed light on that. I should have looked it up.
If bacteria
that's
wouldn't be viable without it.
Yeah. So, all right. So they show this in vivo. They need the inflammasome to have this coagulation happening. And so one of the things that they ask next is, so where is this coming from? What cells are really important for this whole process? And my favorite. They deplete macrophages. So, it's a sledgehammer approach. They treat with clodronate liposomes and deplete all the monocytes and macrophages. And they show they can do this in their supplementary data. And what happens, again, looking at prothrombin time, fibrinogen, TAT, survival, pretty much if they deplete the macrophages, all of those factors look like they do in the untreated animal. So they all go away. So somehow the macrophages are responsible for contributing to the change in the coagulation capacity, which I think is really cool because not very many people that I know of study coagulation of macrophages, although my lab is starting to do that, which is kind of fun. Before the paper came out, before I read this. But yeah, so I think it's really interesting. I had never thought of macrophages as being inducers of coagulation, but they are in fact a key source of tissue factor and other coagulation activity in the blood. So for whatever that's worth.
So that's independent of blood vessel.
That's right. They make their own tissue factor and they can induce coagulation.
And it's produced in the cytosol? Well,
so that's part of what they look at, right? So where does this stuff come from? Are they just what we have to remember is that tissue factor is a membrane protein that gets cleaved, right. And so macrophages could have it on their surface. They do, and we've seen it.
Spoiler alert.
Or it could be packaged in some sort of vesicle or some mechanism like that in order to be able to be released outside of the cells. So it could be either way. What was I going to talk about next? One of the things they did was there's a possibility that there was a role for eicosanoids in this whole thing. So it might not be directly with inflammasome, but it could be indirect. And they used aspirin to block that. And there was no effect on the EPRJ induced coagulation. So it's not that mechanism. But what mechanism is it? So macrophages are known to do this pyroptosis thing. So that's where this comes in. And they wanted to know whether this type of death might be contributing to this whole process. So this pyroptosis is mediated by this protein called gasdermin D. And so gasdermin D is the protein that gets cleaved and activated in the cytosol that will now form those pores and allow IL-1 and IL-18 out. And so a simple way to ask what's going on is just use gasdermin D deficient mice, give them this EPRJ protein, and then ask whether or not they get these same sequelae of the disseminated intravascular coagulation. And turns out that they don't. So it's very important for this process.
Right.
So they were able to show that if you lack gasdermin D, you don't get secretion of tissue factor, cleavage, and activation of caspases, which presumably follows that you don't get IL-1 and IL-18 released, although they didn't look at that. And also that microvesicle release with tissue factors in the blood and by other cells also happen in a gasdermin D dependent way.
Woo.
Yeah.
Mouthful.
So it's a really nice mechanistic paper how they're looking at all of this.
Yeah,
so this is happening in macrophages.
This is happening in macrophages, right?
So this EPRJ is getting to macrophages, activating, and you're getting the pores formed.
Yep.
And out comes
tissue factor,
and also your ILs, right?
Right.
And it doesn't matter in any other cell because when you take out the max, the mice are fine. So even though EPRJ may be getting into other cells, it doesn't matter.
It doesn't seem to matter, right? It seems to be important that the macrophages are doing this whole thing. Yep.
It's interesting. And I wonder, because macrophages are a tissue resident cell, so then, depending on what tissue would be interesting. Because as we've learned from previous episodes, that macrophages are pretty heterogeneous.
Yep.
And so I'm curious, are all macrophages the same?
So I don't know. I know we've derived macrophages from bone marrow, from mouse bone marrow, and they have tissue factor in the coagulative.
Right.
I think in this case they're looking primarily at blood monocytes and macrophages that are circulating to get into their tissues or whatever.
Right.
So I'm not sure what role that tissue resident macrophages play in this whole process. I'm not really sure. I know that when they did this depletion, they did clodronate liposomes IV. So they're mainly depleting circulating cells.
Right. That makes sense. And we're talking about, blood coagulation. I'm just thinking about the effect, because, when you get rid of, like, did they get rid of any of the tissue resident ones? But probably not. It must have just been the blood monocytes that then they're deriving macrophages from. So I don't know. It's just interesting when I was trying to work that through, what the other macrophages were up to and if they have this tissue factor in their plasma membrane. So something to think about.
Yep. And they did a couple of other things where they showed that this wasn't just unique to this EPRJ protein, but that other bacterial proteins that get injected in also use this process, that it also happens in human cells and not just mouse cells. And they did a couple of other things. So I thought that part was really cool. They also showed that they connected this together by showing that the tissue factor that is released from the macrophages that's circulating in the blood really is responsible for that DIC, because then they were able to block it. So they used a blocking tissue factor antibody, and using that antibody protected the mice from death and reversed some of the changes in the coagulation parameters. So it reduced the changes in prothrombin time, the TAT, and improved survival. So again, summarizing, you're looking at the recognition of this bacterial protein that's normally injected in the cytosol is somehow injected into macrophages or monocytes, and that this activates the inflammasome. The inflammasome activates the gasdermin that puts holes in. It also activates IL-1 and IL-18 that get released, and also allows release of microvesicles that express tissue factor. And these microvesicles that express tissue factor then go on to induce this coagulation in the animals, and it's lethal.
Did they try any cells in culture, add EPRJ to it, and see if anything happens?
I thought they did a couple of experiments at the very beginning in their supplementary data, where they just showed that if they added it onto the macrophages, they induced this caspase-1 activation.
Right.
But not other cell types like a fibroblast or an epithelial cell.
They didn't look at those.
It'd be interesting to know if it doesn't happen, why? What's, because presumably the EPRJ will get into those cells.
Right.
Especially with the anthrax part on it, right?
Yep.
So then why don't they respond? Maybe they're missing something.
So maybe they don't express the right NLR or NOD-like receptor. So this one requires NLRP3. I always forget which one it is.
Yeah, NLRP3, pretty sure. Or these are the only cell types that have the tissue factor. We'd have to look at you just look at fibroblasts or epithelial cells to see if they also have these microvesicles.
They might produce IL-1 and IL-18, but not release the tissue factor containing microvesicles.
Right.
That's possible. So that's why they're not contributing. That could be. They also had one other, I forgot to mention, they also had one other system instead of just using the blocking antibody. And I thought this was cool. So tissue factor is hard to work with for a number of different reasons, but there's no knockout mouse, because if you can't clot, you're in trouble. So there are hypomorphs, so there's mice that express less, but they were able to generate a tissue factor inducible knockout mouse. So when they have tamoxifen inducible CRE, that will then cleave out the tissue factor, and so they can induce the knockout in an adult mouse and then inject. And so basically it acted the same as the blocking antibody, where it reduced the activation of coagulation markers and increased survival. That is like the main part of what they talk about. The other things that I thought were kind of cool were that, and I don't know why they tagged it on at the end, but I think it just draws back to the sepsis with gram-negative bacteria, is that LPS injected into mice will also induce coagulation. It's not using the same caspase as this EPRJ protein, but it's using a different caspase. So it's also using inflammasome, but it's a different effector to induce the same process. And so this is called caspase 11. And so the caspase 11 activation really does require this two-step process. So they prime and then activate with LPS. And so they take this into an in vivo model, and they show that if they prime the mice and they prime them with a different TLR ligand called poly-IC that binds to TLR3, if they prime with that and they come back and they inject with LPS, they now see all these change in the same changes in coagulation parameters. So increased prothrombin time, decreased fibrinogen, increased TAT, and lower survival. And this was independent of TLR4 completely. So it's saying it for sure is some sort of cytosolic sensor. And it's dependent on this caspase 11, also dependent on the Gasdermin D, which is creating those pores, and dependent on monocytes and macrophages. So overall, and LPS is also known to induce tissue factor expression. So again, it looks like when you have recognition of microbes, independently of what they're doing to induce inflammation, they are also inducing this pyroptosis process, which releases tissue factor and is responsible for this coagulation and death. And for the longest time, we thought this was a byproduct of what was going on during infection. But the fact that it's induced in response to a microbe suggests that it really is an ancient defense mechanism to induce this coagulation and trap these microbes during this process.
I think that it's important to point out that death is not always the end.
That is true.
Right.
Right.
Because the way they portray it, if you look at their last paragraph,
death.
The release of this into the tissue factor into the blood triggers blood coagulation resulting in organ damage and lethality. Well, probably not all the time. It probably works a lot of the time to clear the microbe.
One would think,
but we would see the
That's
patients who die and
Right.
or the mice that die. But the point is, this is probably working.
It probably is. Yeah.
Right, a lot of the time, and those people they survive. Blah blah blah. So I don't want people to think that this is you're just going to die if you get this sepsis. Right. Because I think it wouldn't make sense to kill the entire organism all the time. And the other issue is I bet there's a range of responses depending on the sequence of the genes encoding all these proteins at all the steps that we've talked about. You have people who are over exuberant, right, in their responses.
Absolutely.
And others who are pretty restrained and maybe don't die. So it's pretty subtle.
Yeah,
right?
I think that probably what the takeaway here is that it is a defense mechanism that just like any other response in the body can go too far or be unregulated. Right. And that when it's working properly, you probably don't even notice it. But if you have a systemic blood infection that's overwhelming, then you end up with this happening a lot. And that's problematic. If you throw a couple clots here and there because you're trapping a bacterium and then you degrade the clot, which we didn't mention that at all. So clot's not a one-way thing. When you cut yourself and you get a clot, eventually that clot scabs over and then it starts to go back to looking like normal tissue after a while and the scab falls off, right? Well, that's because there's reorganization and repair of the tissue underneath. So just because you get a clot doesn't mean it's just there and always there. It's continuously remodeled, and we're always trying to strive to go back towards homeostasis, to back towards the way it was originally. And so if you get these little clots thrown in your body, they're going to trap the bacteria, maybe the bacteria get eaten, and then the cells that are there start to degrade that clot and everything is fine and you never even notice. But it's just something that we wouldn't have necessarily considered this as a defense mechanism.
Right.
And I just think that's the cool part of this. Like, I hadn't thought about clotting as a defense mechanism.
Sure. Yeah, I did not. Absolutely.
Right. Me too.
Totally novel way of looking at it. So I have a paper here that I've been thinking about for TWIV for a long time. It's from Nature. It's called Nlrp9b Inflammasome restricts rotavirus infection in intestinal epithelial cells.
Oh my gosh, I just posted that in our notes below that cascade. Let's check that out. That's funny.
I have, but yeah, so this came out 2 years ago, and it's been in my TWIV potential folder for a while. And because I didn't do it because I didn't know anything about inflammasomes, but now I know more.
Now you know.
And maybe I'll do it. But this is the story here. In this case, this is a intestinal epithelial cell specific inflammasome, an Nlrp9b, and when the virus infects those cells, the RNA is recognized, and it causes maturation of IL-18 and gasdermin induced pyroptosis, and this protects the mice. Although they don't go beyond that, it would be interesting to know what comes out of those pores and how it's protective and what's the mechanism.
Yeah,
right?
Sure.
So
it's very cool. You should do that this coming Friday. That'd be like a paired episode with ours. Drive traffic.
I should,
way.
but I'm so efficient. I already got the papers picked.
Oh, okay. Maybe next time.
I could do it on an immune too.
Yeah,
sure.
I do like to do virus papers on this show.
There's a theme.
Yeah,
there,
we do.
yes.
We do have to go a little out of our comfort zone sometimes, though, because I keep doing innate immune papers, and you keep doing virus papers,
and I keep doing like antibody adaptive immunity papers.
Yeah, I don't know. I figure you would help me out if I got in trouble, both of you. But so I don't know if it's a matter of comfort zone. It's just what you've been thinking about your whole career, right? You know?
Yes. I'm trying to pick things that I'm interested in, but that have a different bend, a different way of thinking about immunology than I normally think about.
Yeah, for sure.
Yeah.
So Cindy, with this paper, I'm curious, I don't know. I'm not a clinician, so I don't know, when people get DIC, I mean, is it just a death sentence, or can you try to stop this rapid clot formation they use, I'm assuming, the same type of, medications that people take, but on greater doses? So I'm trying to think of ways that, what we learned from this, can we target something differently? Can we target the inflammasome? Can we target, it seems like it would be challenging to do that, but did you have any thoughts for, down the road applications?
Well, I'm certainly not a physician that treats sepsis or a physician at all. But
people can write it if they have thoughts.
But I've heard people talking about this, that, these downstream sequelae of sepsis are one of the most significant medical problems that you have to deal with. Vincent's right. Not everybody dies when they have sepsis, but it is often a major problem. And once you get to this clotting, it's a real problem.
Right.
And as far as I understand, and talking to some of my clinical colleagues, there's two stages. So first, there's a hypocoagulative state, and then there's a hypercoagulative state. And one precedes the other. And if you can use one as a biomarker, and we now understand some of the mechanistic aspects of how the second part goes, maybe there's an opportunity to intervene before you get to clots everywhere that you're going to have hard time handling.
Like if you can pull out a certain threshold of tissue factor containing microvesicles, that could be your biomarker.
Right.
Right.
Potentially.
Shall we do some email?
Sure.
Cindy, do you want to take that first one from Pete?
I will. I've been talking. Get sick of my voice, but okay.
Never. We never get sick of your voice.
So Pete writes, Dear Immune hosts, I've been a big fan on the TWIV podcast dating back to TWIV when I was just starting my postdoc in 2009. As a trained but no longer practicing immunologist, I'm so glad to see a podcast dedicated to the host side of host pathogen interactions. I listened with great interest to episode 17 as my PhD involved dissecting the molecular mechanisms of immune suppression of tumor-associated myeloid cells, example, myeloid-derived suppressor cells, which many consider a flavor of tumor-associated macrophages. I just wanted to add a postscript to episode 17's discussion on tumor-associating macrophages and the eat me, don't eat me signal. The potential to disrupt these pathways have not gone unnoticed to industry, as a couple of companies are bringing anti-CD47 monoclonal antibodies to the clinic, including Surface Oncology and Trillium Therapeutics, among others. Of note, Surface Oncology's lead asset recently stumbled in their phase 1 study. The company scaled back the expected enrollment of their trial due to dose-limiting side effects. It leads one to wonder if it could be due to broad CD47 distribution in the body that was noted during your discussion during the program. It will be interesting to see how others fare as they enter this space. Of note, Trillium has engineered multiple versions of their CD47 monoclonal antibodies to reduce the antibody-dependent cellular cytotoxicity activity of the FC portion of their antibody, potentially a way to reduce off-target cytotoxicity. On a more self-serving note, well, before I go there, I think that this is an excellent point. I think we had a really nice discussion about how CD47 mediated internalization and signaling was resulting in the generation of myeloid-derived suppressor cells and suppressing the inflammatory response, and that's definitely related to tumors. So that was really nice. Thank you for that. And I hadn't realized that was in clinics and had run into some difficulties. So maybe our discussion will lead somewhere if somebody hears this. Okay, so continuing. On a more self-serving note, since leaving the bench, I've gone on to consult for biotech and pharma companies, focusing on helping them think through the clinical, scientific, financial, and strategic issues that underpin their business model. Given the clinical and commercial success of checkpoint inhibitors and the subsequent explosion of investment in immune oncology approaches, this topic has been a large part of our practice over the past 3 to 4 years. As a result, we often work with companies in the early stages of formation capitalization and see a lot of exciting emerging technologies and approaches at the interface of the immune system and cancer. To that end, my colleagues and I recently published 2 white papers on areas at this interface that I find quite interesting, namely the concept of cancer-associated neoantigens and the impact of the microbiome on immunotherapies. I think you would find both intriguing, and I've included the links for your perusal below. Look forward to many more episodes to come as it helps me keep up with the field on my daily commute. All the best, Pete. So I guess we can put those links in the show notes if he's okay with us sharing them. I think that's great. I think this is really cool that I always thought, consulting or this is going to be a boring job. But you know what? Maybe there's a benefit to that, seeing all the cool things that are coming out and see some of it go actually to potentially get approved. We're so far removed from that when we're at the bench doing the basic immunology studies. We're so far removed from the translational aspects of it sometimes. You don't realize how exciting that could be.
Yeah, I think I was talking to somebody who went into industry directly after their PhD, and she's loving it. So I think it's kind of a difference in how do you want to work on the more basic side? Do you like the translational side? And then either side probably thinks the other side is boring a bit because, similarly, I was like, oh, that might seem monotonous, but you're right. Especially with immune oncology therapies, it's just such a hot area.
Exploding. So. Yeah.
I think everybody's different in what they like, right? So what seems boring to you is not to someone else. But this whole idea of consulting, I mean, this individual sounds like he was in the industry for a while. Yeah. That always struck me as the way to do it. You get experience after so many years. I don't know how many years it takes, right? And then you can advise people on what to do. It always struck me as weird that you became a consultant right out of your PhD because what's your knowledge base, right? So, but I mean, it could be different where, if you're in the financial sector, the people who have no science background at all can say to you, explain this to us And so we can understand the potential. So that could work.
Right.
But my wife, for example, after 35 years in pharma, she's a consultant now, and she advises people on how to get the preclinical data they need to get an IND, an investigational new drug application. And she wouldn't know that after 1 year out of a PhD, right? So there are different kinds of consultancies. And I think the ones where you're advising on what experiments to do really need more of a experience base than other kinds. That's the way I look at it anyway.
Yep. Yeah, maybe Pete, you should write back in and tell us kind of how you got into it, how you see our conversation. And because there's a lot of people who are interested in this area that don't want to continue with academia, want to go into industry. So be some good insight.
Steph, can you take the next one?
Yes, sure. Cindy, Steph, and Vincent, I just want to say that I thoroughly enjoy listening to Immune. I began listening to TWIP recently as well, since I enjoy parasites, and I'm doing research on, Vincent, do you know how to pronounce this?
Naegleria fowleri.
Perfect. A brain-eating amoebic parasite. I'm also emailing you for something that I think is very important to the listeners like me that are not to grad school yet. I'm finishing my undergraduate at Weber State University, and I'm beginning to look at PhD programs to apply. Since this podcast has what I would say are the 3 stages of academic careers, with Steph being in the beginning of the career, Cindy being in the middle, and Vincent being the final steps of a career. Although I don't know, Vincent, if that makes you feel a certain way. We don't have to say final. We can just say the latter part. I want to see what your advice would be for those of us who are just starting. What is your advice for trying to find the right graduate, master's, or PhD for people to apply for and to figure out what they want to study and research? Best regards, Weston Hutchison. And I think this is an excellent write-in, always a topic of conversation. So we could do like a round table, answer.
So what's the question? Trying to find the right program for people to apply to?
I think his question is, how do you prioritize what you should be looking at? So whether that be the mentor relationship, whether that be the research topic, whether that be location, is that person senior, they're publishing a lot? I think, how do you prioritize that when you're looking? Unless you guys see it differently, that's kind of how
I can pipe in a little bit at the beginning. The advice I give my students who are heading out for things like this, the undergraduates that I mentor, is find a place where there are multiple people who are doing research that is interesting to you, and that they are doing high quality research. Because after that, you'll probably be okay. Right. So as long as there is a good, solid program with a lot of individuals who are doing research of interest to you, one of them will pan out once you come there. Because a lot of programs are designed so that you're accepted into an umbrella program. And then within that, then you rotate in different labs, and then you choose based on the person's lab that you rotate in. If they like you and you like them and the fit is good, then you stay there and do your PhD. And it has to be a combination between the topic that you are passionate about and the mentoring that you'll get. Because I think if you choose a lab where you're like, I love this topic, the PI sucks, but I know I can do this, that's a recipe for disaster in a lot of cases. And the flip side is true too. You're like, I really love this person as a mentor, and the research is okay, but at least I'll learn something. It's also maybe not the best situation. So you kind of have to test out the waters and see what fits. I don't know what you guys think.
Yeah, Vincent, you can go first.
I think the most important thing you can do is find the right mentor and listen to them.
Yeah, that too.
And it's not easy to do, but because you're going to have to listen to them. And if you're going to find a mentor and not listen, there's no point in having one.
Right.
And so it almost doesn't matter what you're doing. I mean, if you're interested in life sciences, you have a lot to do, a lot of different choices. I think that's secondary to finding a great mentor. And you have to talk to people. You have to get advice from others who are at the same institution. But if they're in the area that you're interested in, certainly your interest should be a driving force, your interest and curiosity about the subject. But you have to find someone who's a good mentor, because you can have the coolest project, but if the person's not a good mentor, it's not going to work out for most people.
Yeah. So that would be my number one as well. I think you really have to be, forward thinking, almost in an aggressive way. You're trying to decide, who you're going to be spending the next 5 years, give or take a few, depending on your program, of your life with. You're really working with this person, this mentor, very closely. So I agree. I agree with both, but finding a mentor and talk with the graduate students. If you're going on an interview and you do not get the opportunity to talk to the lab members one-on-one without the PI being there, that's not a good thing. I think you really want to get the perspective of the people doing the work every day to get an idea of what the dynamic is like. Because you're right. If you have a good mentor, at PhD, you're in training, you can always, if it's not the perfect field for you, if you have a good basis of training, you can take those skills to study a different virus or study a different immune component. But if you don't have that background, that training, and you don't have a good relationship, it could be a struggle to after the PhD to really land on your feet. So I would say mentor first, but the close second would be that research aspect. So I agree with both people.
So let me say something about this final steps of a career.
Yeah, I didn't know how you felt about that. That was his words, not mine.
Well, I don't know how Weston would know either way, right?
I think he means ladder, if you're considering
because some people, guy at Rockefeller just died. Yep. You must know Paul Greengard.
Yep.
He's 90 years old.
Yep.
He had 60 people in his lab. Yep. He was obviously going to go another 10 years. He wasn't even thinking of being in the final steps. So, I don't think Weston, maybe I've talked a little bit about another, but so I'm not offended whatsoever, but I don't know what it means to be in the final steps of a career in science because it could go on forever. I have no desire to go fishing. Okay? I have no desire to do anything but think about science. Okay. And so I don't consider I'm going to have a change in a career, right? I'm going to probably do communication.
Right.
For, once my lab is closed. So you could say I'm coming to the the end of the first phase of my career.
That's what I was thinking.
Yeah.
And then do something else because I can't imagine not doing anything. I know a lot of people like to travel and eat and read, right? But I do that anyway for science.
Exactly.
So I'm not going to just do that. And so I view it as a second career. Yeah, and I think most people in science don't have second careers, so I'm pretty fortunate to be able to do that. And by second career, I mean science communication.
Yep.
All right, one more from Andrew. Hello, Immune Team. The last two podcasts have increased my curiosity about how vaccines are made. I understand the basic premise of generating an immune response with attenuated vaccines or others. What I don't fully understand is large-scale vaccine production and the function of additives such as aluminum. I'm specifically curious why aluminum is needed to enhance an immune response and why an attenuated virus is or is not enough to generate a strong enough response. Thanks for the info and carry on. So, Andrew, I suggest you go over to my YouTube channel and listen to the lecture on vaccines, youtube.com/PROFVRR. You'll find the 2019 virology course there. There's a lecture on vaccines that'll answer some of your questions, including an attenuated virus does not have an adjuvant.
That's right.
At all.
Right.
Because they replicate. You don't need to have an adjuvant. Some non-replicating vaccines, inactivated virus-like particle vaccines, they don't replicate. They don't cause cell damage. They don't cause inflammation. You don't get a good immune response. So we add adjuvants like aluminum salts, not aluminum, which is a metal, right? Aluminum salts, or other things, TLR agonists, to stimulate inflammation. And these include the Hep B vaccine, the human papillomavirus vaccine, and others. But interestingly, the inactivated polio vaccine, which is inactivated, it doesn't require an adjuvant. It's good enough on its own. So it's not a universal thing. And no attenuated infectious virus vaccine requires an adjuvant. And because they replicate,
right,
you don't need to have one.
Right.
And often I get into arguments on Twitter. about MMR, measles, mumps, rubella vaccine.
Yep.
They want to know, why is there aluminum? And there isn't. There never was, and there never will be.
Right.
Get very worked up about vaccines because people listen to the wrong people.
That's right.
Going to be my pick of the
Yeah,
week
you want to go
today.
first?
Good transition.
You know, this woman on Twitter the other day said, I don't believe anything you say.
What?
To me.
Oh, boy.
And I said, well, science is not a belief system.
That's right.
The facts are out there. Just go read the literature.
That's true.
Yes.
My pick of the week is a book by Paul Offit. It's called Bad Advice, or Why Celebrities, Politicians, and Activists Aren't Your Best Source of Health Information. Now, Paul Offit is a pediatrician at Penn. He has published a number of books about vaccines, including Autism's False Prophets, another one which I recommend to people who want to know basic information about vaccines. It's called Vaccines and Your Child, Separating Fact from Fiction. It's great. It explains how vaccines are made, what's in them, what they do, and debunks all of the nonsense that's out there. And I have to say, pretty much everything negative you've heard about vaccines out there is nonsense.
Right.
We wouldn't be using them on hundreds of millions of people for 50 years if they had a fraction of the issues that people say they do. I just don't get it. The lady on Twitter said, my son never spoke again after getting his vaccines. Really? And so if you tie your shoes and it rains, does that mean tying your shoes causes it to rain?
Right.
So people don't understand that when things happen at the same time, it doesn't imply
Causation.
Right.
So this new book from Paul Offit, it's about a year old now, but he says scientists don't like to speak about what they're doing. They're not public speakers, and so we have celebrities and advocates, lobbyists, funders who take advantage of scientists' reluctance to provide easy answers and flood the media with misleading or correct claims about health risks. And so this is a book all about that, and I think it's really good because he writes well and he knows the problems with non-scientists talking about science, and that's been my shtick for years now.
Yep.
I want scientists to talk to the public about science because they know it the best. Everyone else, there's some great science writers out there and so forth, but he's talking about the charlatans out there who are not the right people to give you advice. So I think I'm reading this right now. It's really good. So I think
it's on
maybe
me
maybe
too.
and also the other one, if you want to, and if a friend asks you, what can I read to explain vaccines, send them this Vaccines in Your Child. It's really great. It's great for the general public.
Awesome.
There you go.
Cool.
Steph, what do you have?
Yeah, I have. So there's this series of, it spans really all of biology. It's called iBiology, and there are videos by the scientists themselves. So reiterating what Vincent says, about their topic, and they give you a lecture on their specific, specialty. And there's called Immunology Flipped course. And basically it's a graduate level introduction to immunology. It is free, and it allows you to watch these videos, learn more about immunology. And if you are an educator, you can actually sign into their kind of platform and get assignment materials. But I went through a couple of them. They're really good with some of the really big figures in the immunology field. So check that out if you're if what we provide, if you're still having questions, which there's a lot to talk about, it's a good resource.
I'm glad you picked that one because the former chair of my department and my colleague, Avery August, did one of those on allergy.
Yes, I watched that one. Yep. It's good. Very good.
Yep. It's a well-known series besides immunology too. Yeah, it's very great.
I didn't know about it.
Cindy, what do you have?
Yeah, so I have a link to a Science Alert article that describes a scientific article that came out recently. And yeah, it's a small article, but I thought it was kind of interesting. The article is about a new study that showed that a conversation with a person who had a vaccine preventable disease was very effective in changing the minds of people who are vaccine hesitant. So what they did in this case was they took, and it was a very select population. There are a lot of caveats to this, but I think it just hammers home science communication and telling a story, right? And so they took college students who were vaccine hesitant and they put them into groups, and they had them either talk with somebody who was a scientist or talk with somebody who had a vaccine preventable disease. For example, somebody who got shingles and, had a lot of pain that wasn't well controlled or whatever, or had polio. And although the study was small and basically it's predictable to those of us who are in this area and get it already, but I thought it was interesting. But they showed that 13 out of 19 of the students that were exposed to, a conversation with a person who had experienced a vaccine preventable disease became much enthusiastic to vaccines and were no longer vaccine hesitant. So, it's not a huge series of facts that really changes people's minds, but rather the stories. And it makes me happy and angry at the same time, because it's just like, why do we have to go to that length for people to believe us? It's like, you have to actually, what's in it in human nature that you actually have to experience something to believe it? Right.
So, and the problem with that is with vaccines, we have less people who have gotten those diseases. And so, if it's doing their job, then there won't be anyone to tell those stories. So it is frustrating.
So it's people say that vaccines are the vaccine hesitancy is a product of the success of vaccines, right? They've been so successful that people don't see the diseases anymore. So therefore, their interpretation of their level of risk changes. It goes down. They don't think it's risky. So they think the vaccines are riskier than the disease, but they just don't understand the concept of the reason why we don't have these is because of the vaccines. And if we back off, they're going to pop back up and people are going to get really sick again. And I actually wrote an opinion article about this that was published on CNN, and I called it Societal Amnesia of Infectious Diseases, Childhood Infectious Diseases. And it was just that idea, it's like if people don't see it, they forget about it. And so while our parents still had instilled in them, mine anyway, we're different generations here, but mine knew that, their friends had polio or whatever. They didn't want their children to suffer. So they were really insistent upon taking advantage of every vaccine that was available. But then the moms now have this luxury of saying, well, it's not that risky for my kids, so I'd rather not vaccinate them. So it's but what do you do? So we can't keep pulling these people out of the woodwork to tell their stories. We can try, but if you don't have that face-to-face communication, how else do we get that story across to these people to make that impact? That's where I think that the research in this area needs to go.
Right.
It is immensely frustrating that we have to go through this.
Yeah.
After 50 years of vaccine success and eliminating in many countries huge numbers of serious infections, that we actually have to justify this because people don't understand how they work in the relationship between things that happen and causation.
Right.
But what's even worse is they don't want to listen to scientists. And they don't believe us.
Right.
They listen and believe to the charlatans who are preaching the negative view of vaccines like Andrew Wakefield. There was a big rally, in Rockland County, right? There's a measles outbreak.
Measles outbreak, yep. Mm-hmm.
And, they've introduced legislation to prevent kids under a certain age from being out and about unless you're immunized. They had a rally a couple of weeks ago. The anti-vax people had a rally. They attracted hundreds of people spewing nonsense. They had a rabbi get up there and say, it's better to get naturally infected because it reduces your chances of heart disease, cancer, and some other disease, diabetes. I don't know what.
There's
totally
no
preposterous.
No studies that support that.
Totally preposterous. Yet people hear this and go, "Oh, yeah, that's why I don't want to vaccinate my kids." But they're not even critical about it.
Well, and there is a legality of basically promoting something that could cause people true health risks? What is that's a real I mean, you're actually could damage people. I mean,
absolutely. Absolutely.
Mm-hmm.
States have the right to protect their populations from infectious diseases. This is constitutionally acknowledged by legal scholars. Okay. States have the right to say you have to be vaccinated.
Yep.
And it's going to come to a point where some really vocal anti-vaxxer is going to cause a huge outbreak. It's big enough as it is now with measles. It's going to be a huge outbreak, and they're going to be liable. Somehow we have to do that. We have to sue these individuals for causing outbreaks because it's totally unacceptable. This is not where we need to be spending our time arguing. Vaccines work.
Yeah.
I've wondered
that opens
if
a whole
there's
can of worms.
Yeah, I've wondered if there's a way to shift the conversation. So, for example, when I've talked about climate change with people, if they start arguing with me, it's not man-made, it's not happening. I'm like, okay, whatever. Whether you believe it's happening or not, why do we want to destroy our planet?
Right.
Right? They can't argue with that. It's like, maybe I believe that those actions that people are having are man-made, changing the climate, but regardless of whether those second two things are true, we are destroying our planet, whether that's leading to anything or not. Why do we want to do that? Right? And so if you can shift the conversation to something that everyone agrees on, like you disarm them, right? They say, oh, well, yeah, it's actually a bad idea to litter and to put things into streams and destroy the planet and kill, make animals go extinct and whatever. I agree with you on that. If they agree that, and then it's like, you don't need to go any further. It doesn't matter if you agree with me on climate change. It doesn't matter if you believe in climate change, don't believe it's man-made. None of that becomes irrelevant anymore if you can find a common ground to agree on. And so I've been trying to think about how can we shift the conversation about vaccines to something that people can't argue with. And they will be like, oh, well, yeah, you're right. And I haven't figured it out yet, but I feel like that would be something that we could use. And then we wouldn't have to spend all our time defending, because basically this is a tactic in oral communication, right? Where you constantly put up barriers that someone has to defend and they never get to make their actual point. Right. Right. So we're, as scientists, as a society, as communicators, we're spending all of our time defending all of the crap that comes out, and that's just taking up all of our energy, and we're not using it to communicate the things that would be more effective. But I haven't figured out how that would be yet.
Yeah, I was going to ask you, what's the angle for vaccines?
I wish I knew.
Yeah. We do have a listener pick from Steve. He writes, here's a book pick for Immune. I'm only a couple of chapters into it, but I already recognize many of the terms, having heard them on Immune. So this is a book called An Elegant Defense, The Extraordinary New Science of the Immune System, A Tale in 4 Lives by Matt Richtel. I don't know anything about it. Has anyone ever heard of it?
I have not heard of it.
No,
okay,
I haven't.
So there you go. It's a recommendation from Steve. Thank you, Steve.
Yeah. Thanks. Let us know when you finish it.
That's Immune number 20. If you want to hear, if you want to read the show notes, which means the links that we put up and our picks and so forth, you can go to microbe.tv/immune. If you listen on a phone or tablet, a podcast player, please subscribe. It lets us know how many people are listening out there. That's an important piece of information for us. And if you really like what we do, go to microbe.tv/contribute. You could, for example, use either PayPal or Patreon to give us $1 a month, less than a cup of coffee, for wonderful science. And you not only get Immune, but you get all the other science podcasts that we do. We'd really appreciate that. Cindy Leifer's at Cornell University, over on Twitter, Cindy Leifer. Thanks, Cindy.
Thank you. Till next time.
Steph Langel's at Duke University. Stephanie Langel on Twitter. Thank you, Steph.
Yeah, thanks. This was great.
I'm Vincent Racaniello. I'm at virology.ws, Prof VRR on Twitter. Music on Immune is by Steve Neal, stevenealpercussion.com. Thanks for listening to Immune, the podcast that's infectious. We'll be back next month.